and everything. So normal structure of the DNA. But rather than this normal structure, we have a little bit different structures here, which is exceptional sometimes and which is more common for RNA also, because RNA is single-stranded and it has sometimes it has complementary structures, complementary sequences. So this is one is that hairpin structure. When it is single-stranded, they can the complementary strands can bind like this. Because it is single-stranded, they are open. These hydrogen bonds are open. Here are the nucleotides. So these nucleotides, if they will find the complementary structure here, then they can bind to. And as these are hydrogen bonds, these bonds are very flexible, very weak bonds, so they can form this bonding, and when it is required, they can open. So this way, they can create some kind of hairpin structures. If there is no complementary sequence here, then they cannot bind together. So that part is open. And when there are complementary sequences, they can bind together. And in case of RNA, we can find this kind of binding a lot, particularly when we'll go for the tRNA structure, we'll find there are 4 loops there, 4 particular loops. And this is called the hairpin structure. And if there is no such hairpin structure, then what will happen? They can bind together. They can make a total. There will be no loop, but they can make double-stranded structures. So these are a couple of examples of hairpin structure, and this is a stem-like structure, and this is kind of RNA structure. RNA has a lot of hairpins like that because it is single-stranded, and if they are complementary bases, they are tending to join together. Now another thing is that HDNA. It is a 3-stranded structure. So there is one double-stranded DNA there, and another strand is open, but the sequence is matching with the double-stranded DNA. In such case, they can make this kind of structure. This is a 3-stranded DNA. The complementary sequences are there, and they can match. This is very common in case of mammalian genome, because in mammalian DNA, there are a lot of repetitive sequences, a lot of repeats, same sequence over and over, and even for million copies up there. In such case, they can create this kind of structures. And these are not really having so much problem. Sometimes they can create problems because of RNA synthesis and other things. And particularly for that cases, sometimes we could find some disorders or something. But this one is very important because sometimes one methyl group can add to a nucleotide base. If the methyl group is added to the nucleotide base, it is called methylation. And DNA methylation, generally, if it is methylated, that means it is creating some kind of cap. And no other compounds and no other bonding can be done into that part. That is creating a cap-like structure. Methylation is very common for RNA when RNA is produced, because at that time this methylated cap is required. But in some cases, if this kind of methylation occurs in the DNA, and where the cytosine, this is the most common for cytosine only, the methyl cytosine, formation of methyl cytosine, they can create methylated cytosine here. And this is called CPG island. CPG island means a particular area with methylated cytosine. And this is very common for prostate cancer. In prostate cancer, we can find a lot of CPG island into their DNA. We really don't know what is the actual function of that one, but this presence of CPG island is very common in case of prostate cancer patients. So these are a couple of extra structure or extraordinary structure of DNA, and we know about the whole DNA structure. So DNA part is over. Then we'll go for the replication and other things. But now we need to know about the structure of chromosome. let's go for the structure of chromosome. Now, what is chromosome? Chromosome means a threaded structure in the nucleus, and during interphase, we can find a reticulated structure which is called nuclear reticulum. Then this nuclear reticulum coils up during the cell division. During cell division, we can find the chromatids, two chromatids, and a centromere bound to it. That is called the chromosome. Generally, we can color this one by strands. That's why it is called colored body or chromosome. Chromosome means chromo means color, and zoma means body. That's why it is called colored body or chromosomes. Now, this chromosome is made up of DNA. In case of bacteria, the chromosome is made up of only single DNA. But in case of eukaryotes, the chromosome is made up of DNA and protein. Okay. Now the thing is that DNA is very long. It is very long chain. In case of prokaryotes, it is approximately 4.6 million base pair in one DNA. 4.6 million base pair, and they need to be packed into the cell. In case of human, they contain almost 6.4 billion base pairs. And if you stretch one single DNA, it is 2 meters long. 2 meters means just like this table in single DNA. And it is said that if you take out the all DNA from a single human body, you can go to the moon. You can just add it. Now think about that. That 2 meter DNA need to be coiled up in such a way that you cannot see by your naked eye. Okay, the cell, you cannot see the cells by your naked eye. Okay, so this should be tightly packed in such a way. So this one, in case of human DNA, it could be 1400 times packed or coiled, that 2 meter DNA. So this is the main thing. The DNA needs to be coiled up, and the coiling is done in two ways. One is the strand coiling, and after the strand coiling, there are more coiling there. Strand coiling means when the DNA is like this structure, so like this, the bases are here. This is one turn. In one turn, they can have 10 bases. Generally, this is the normal structure of DNA, that they can have 10 bases. Now, they can be tightly packed here. When they are tightly packed, they could have more than 10 base pairs in one turn. Okay? If they have just like this part, it is called over-rotated. So more than 10 base pairs in each coil, that is called over-rotated, and that is called positive coiling or positive supercoiling. If they are a little bit unwind into that part, so just like this one, that is called under rotation. In case of under rotation, this should be less than 10 base pairs in a DNA per turn. In such case, it is called negative supercoiling. And I don't know whether you know about this phone. Do you have the idea about this phone with the coil? I don't know whether in your generation you have seen this one or not. This is very common in 1980s. The phone with the coils, so you can put it there on the top or on the table phone. Right now you are habituated with this single phone, that is cell phone. Okay, so this is a very good example of DNA supercoiling. There are coils here. Again, those are coiled in such way. All those cables. Okay, that is the kind of example of supercoiling and positive supercoiling and negative supercoiling. Now, it is very important one thing. When you are coiling the things, suppose take one thread or strings. That is, there generally 2 or 3 folds there. They are rotated like this. Try to open up one. When you will open up one, just like this is a thread, like this. If you open up this one, then you will create some kind of gap between these things. when you are creating the gap, you will feel the other end, they are tightly coiled there. And you will have some kind of tension there that it should be unwind or it is rotating in the other way. Try this one. If I will find some thread next day, I will bring that one. Show you. So when that kind of tension is generating into the DNA coils, then this enzyme that is called the topoisomerase, what they are doing, they are trying to open up this part, release the tension, so it is automatically unwind in this part, then rejoins that. So like this, they can open up this one, it is uncoiled, then they rejoin that. So this is the function of topoisomerase. Okay, and this function of topoisomerase is required in our DNA replication part. Okay. Now tell me, a DNA molecule 300 pp long has 20 complete rotations. Is it positively supercoiled or negatively supercoiled or relaxed? Positive, negative, or relaxed? What do you think? Why?
Because if it was relaxed, it would have 200 things. So it'd be
So 20 complete rotations means 10 base pair per rotation. So it would be 200 base pairs. Okay. So it is not relaxed. 300 base pairs in the 200 rotation, that means lot of base pairs there in one rotation, that means negatively supercoiled. If it would be 100 base pair in 20 rotations, that means it is positively supercoiled. Tightly packed. Okay, but in case of bacterial chromosome are not protected by any kind of membrane. Okay, they are open in the cytoplasm, but they are actually composed in a particular space in the cytoplasm, in a particular area. That area is called the nucleoid. Bacterial chromosome has a lot of loops like this. They are forming this kind of loops. The coilings are there, then they are forming this kind of loops. Each and every loop, the end part of the loop, open part of the loop, is protected by a particular protein. Generally, this kind of proteins are called strand binding proteins. Okay, during DNA replication, we will see this one. So these kind of proteins are there. They are just protecting the coiling. That it should not be uncoiled or it should not be more coiled there. If some other enzyme is required to coil that one, then they can form these things. So this kind of coiling is present in bacterial DNA. Even in the eukaryotic DNA, at first this kind of coiling is there, then there are more coiling of these coils. So this is called actually supercoiling, how they are packed into the chromosome. And based on this coiling, so at first DNAs are there, DNA have their own coil by double helix. Then after that, they create more coils there, and those coils are coiling again. So there is a lot of coiling there, and that's why they are packed in such a way. Based on this coiling, it is called, the whole structure of the chromosome is called chromatin. Okay, this chromatin is two types there. One type is called euchromatin, another is called heterochromatin, based on the DNA coiling. And in eukaryotic chromosome, there is not only the chromatin DNA, there are other proteins also. I'll talk about that one a little bit later. Okay. So, what is euchromatin? Euchromatin means the area of the DNA which is little bit less coiled or relaxed state, but where the most protein synthesis can occur. If it is tightly coiled, then enzymes cannot open the DNA, because when there is a protein synthesis or during replication, it happens all the time. There is no problem for replication because all the DNA needs to be replicated to make including numbers. Come on. So DNA is like this, or if I put in this that way, so they are bound to each other by hydrogen bonds. During replication or during protein synthesis, these bonds needs to be opened. Okay. Now, if they are tightly coiled, then they cannot be opened up very easily. And all these functions are done by different kind of enzymes. Okay. So where it is relaxed state, enzymes can open up, they can go there and transcribe the RNA from this DNA. So that is the expression of the gene. So in the relaxed part, there are mostly active genes. Active genes means which can transcribe the proteins. And this part is called euchromatin. Now, the other part, which is tightly coiled and highly condensed throughout the cell cycle, it cannot be opened anytime. It is always coiled, tightly coiled, and there is no such active gene in that part, and there is no such replication, no such crossing over, and if there is replication is at the later stage of the interphase, these are called heterochromatin. And in our chromosome, we have almost 70 to 80% heterochromatinized part. Okay. Particularly, what is the evolutionary significance of this heterochromatinized part? And this is very, very interesting topic in molecular biology. That what is the function of heterochromatin? Heterochromatin part, we thought that all the genes from our ancestors, those are there, and those are not in active stage. Only 2% of our whole genome is active. 98% is not active. 97 to 98% genes are not active in our genome. Only 2 to 3% are active. And in these 2 to 3%, we have 21,000 genes. Okay. All those 90, when we'll go to the evolutionary genetics, we'll find that these heterochromatinized part, they contain the genes from apes. And we have some kind of sequence similarities with chimpanzee genome or gorilla genome with other, and those genes are in this heterochromatinized part. When genes need to be active, they need two other particular areas here. One area is called promoter. This promoter must be present. If there is no promoter, there will be no transcription. In heterochromatinized part, those genes doesn't contain any kind of promoter. If there is no promoter, the RNA polymerase cannot attach to that DNA and they cannot transcribe any kind of mRNA. Based on this heterochromatin, there are two types. One is called constitutive heterochromatin. These are mainly the permanent heterochromatin, just like centromere. There is no gene near the centromere, no gene at all. What is the function of that? Centromere part is highly coiled, highly condensed, and that is the heterochromatinized part. There are only the binding site for two particular proteins, that is called kinetochore. And as the kinetochores are bind to the centromere, they can bound to the spindle fibers. So the centromere is the position for attachment of spindle fibers with the help of kinetochore. In Y chromosome, we call the Y chromosome doesn't contain any gene, because most of the part of the Y chromosome has mostly constitutive heterochromatin there. It is permanently closed. So there is no such synthesis of any kind of protein there. Another is called facultative heterochromatin. It is when it is required, the part of the chromosome becomes heterochromatinized. When it is not required, then you can open down. And that is the example of the X chromosome in female, extra X chromosome in female. That is the formation of Barber. Okay, so one X chromosome becomes inactive during that time. So that X chromosome, it is the program of nature. She determines that which chromosome is active, will be activated, which chromosome will not be activated, which one is not activated, that is becoming heterochromatinized in that way. So there will be no transcription, no expression of genes. That is called facultative heterochromatin. Now think about the whole chromatin structure. So the DNA is there. With each and every DNA, there is one kind of protein attached to it, particularly for eukaryotic chromosomes. This protein is not present in bacterial chromosome or prokaryotic chromosome. This protein is called histone protein. But histone is not a single protein. There are 5 subunits there. Is anybody there? There are 5 subunits there for the histones. H1, H2A, H2B, H3, and H4. Of these, histone contains mostly little bit of positively charged amino acids, and those are arginine and lysine. As they contains more positively charged amino acids, the net charge of the histone is always positive. Why? Because DNA is negatively charged. For this attachment of DNA, histone needs to be positive so they can attract the DNA to bind with them. Okay. This code pattern is made up of these 4 histones: H2A, H2B, H3, and H4. They can form the octamer. So it is like this. These are 4, and here are 4, and here are other 4s. They are forming octamers. 8 total components are there. And they are forming this kind of ball-like structures. This is called histone octamer, and DNA is coiled 1 3/4 turn in each histone octamer. Okay. With these histones, there are some kind of variant histones. When we'll go for the replication and mutation, we'll find that these histones, these variant histones can cause a little bit of mutation there. They are having different kind of amino acid sequences. They are not normal histones. Okay. And as these variants, they are assumed to make chromatin more open. If the chromatin is more open, then they are more sensitive to break, they are more sensitive to transcribe, so they can create more protein and can develop a genetic imbalance there. And some kind of other proteins are there, those are non-histone proteins, and they are found in eukaryotic chromosome, but really we don't know their function very well. So the comparison is that between euchromatin and heterochromatin, euchromatin is less condensed, heterochromatin is mostly condensed. Euchromatin in chromosome arms, heterochromatin in centromeres and telomeres. Telomeres, the function of telomere is that to protect the chromosome. To protect the chromosome means it should be tightly coiled, though telomere is containing some kind of proteins sometimes, but not all the times. In case of euchromatin, the sequences are unique sequences. Unique sequences means each and every gene has some kind of unique sequences. But in case of heterochromatin, there are repetitive sequences. A particular sequence repeats for 100 to million times there, particularly in the telomeric part. There are a lot of repetitive sequences. Presence of genes, euchromatin causes the most genes. Heterochromatin are very few genes. Either it could be transcribed or cannot be transcribed. They are just there. When replicated, this is through S phase. That is the main part. The S phase is the replication phase of the whole DNA. But euchromatin part is replicated fast, and the later S phase, the heterochromatinized part is replicated. Transcription is more often in the euchromatinized part. In heterochromatin, if there are some genes which are active, then they can transcribe, but it is infrequent. Crossing over is more common in euchromatinized part because it is more open, it is sensitive to break, but heterochromatinized part, it is tightly coiled, so there is no such break there. What about this? If we neutralize the positive charge of histone protein, what will happen?
Either B or C.
Yes. Yes. Okay. So now we know that DNA, that means that is the chromatin, chromatin has Z-chromatin, heterochromatin, but that is a thread. And the histone proteins are there. These together are called nucleosome. A nucleosome is the simplest structure of a chromosome. One unit of chromosome, we can say that in such a way. One unit of chromosome, that is called the nucleosome. This is the core particle, is the histone octamer containing H2A, H2B, H3, and H4 histones there. It is creating this kind of histone octamers. DNA is coiled 1 and 3/4 turns onto this, and this DNA has a particular length here, generally 145 to 147 BP long. This is the whole coiling. That part of the DNA is 145 to 147 BP long. And BP means base pair. Okay? What is base pair?
It's like they're the opposite sequences that are like attracted to each other.
Why DNA is measured by base pairs?
Link them together.
So that means we are actually we are measuring the bases. As DNA is double stranded, that means this is called base pair. This is one base pair. So if it is two base pair long, that means actually two bases long. So how many bases are there? That's why we are measuring base pairs, because DNA is double stranded. Okay. So one round of DNA about the histone protein is 147 base pair long. Okay. But histone 1 is not the part of this. It is like the clamp. So this histone protein is there, DNA is coiling about it. Then there is one clamp on top of that to secure this structure, and that is the function of histone 1. So H1 is always outside. This is H1, and this is the histone octamer, and here is the DNA. Okay, so histone 1 is just clamping that one, and histone 1 binds to 20 to 22 BP of DNA. So the total structure of the nucleosome is 147 BP plus 20 to 22 BP extra. Now, what is chromatosome? So nucleosome is this part. When histone is attached to the nucleosome, histone 1 is attached to the nucleosome, that is called chromatosome. So what is nucleosome? Histone octamer with the DNA coiling onto it. That is the nucleosome. Nucleosome clamped by histone 1, that is called chromatosome. Okay. So we know that 145 BP of DNA is required to coil the histone octamer, and 22 BP extra is required for histone 1 clamping. Altogether is around 167 BP long. So one chromatosome, you can find 167 BP of DNA. In one nucleosome, you can find 145 to 147 BP of DNA. Okay. Now each and every histone, we'll discuss this one later. during the mutation class, the histones are not only like this, not like this. They have some kind of protrusions. Like this. These protrusions, this is a ball-like structure, okay? But they have some kind of finger-like structure around it. These protrusions are called histone tail. The function of this histone tail is that they are positively charged. They can anchor the DNA very positively, tightly associated, and they can have the interaction with the other histones to bind with each other. Okay. If these protrusions are less, if these protrusions are long, then they can create some kind of structural problem in the DNA structure, in the chromosome structure. Okay, so we have the nucleosome and chromatosome. Up to this one is chromatosome. If one chromatosome is attached with another chromatosome, so here is another histone octamer, DNA is coiled here, histone 1 protein is there. Between these two, this DNA part, a little bit stretched DNA part is there. Between these two chromatosomes, that is called the linker DNA. Okay, and this linker DNA is about 30 to 40 pp long. And all those non-histone proteins, if it is there in the chromosome, they can be attached to this linker DNA only. Because the histone nucleosome part, it is very, very tightly packed. So there is no way that other things can go to that one. Now I think we have the structure. Okay. So this is the core histone protein, the histone octamer. And each histone octamer, it is almost 11 nanometer long, and DNA is coiled here, that is 145 to 147 pp of DNA, and is coiled about 1 3/4 turn, approximately 2 turns of the DNA. Between these two nucleosomes, right now the histone 1 is not there, so these are called nucleosomes. Between these two nucleosomes, this part of the DNA, this is called the linker DNA. Okay, so this is the very basic structure of chromosome. Now, up to this part, the thread is prepared. So DNA was there, nucleosome is there, and the whole thread is prepared. This is called actually chromatin structure. And there is another name. Now this whole thread, this beaded structure, so histone DNA, and histone DNA, this beaded structure, they need to be coiled more. Like the positive supercoiling, the negative supercoiling, like that. In such way, they can make the higher order chromatin structure. First, nucleosomes fold themselves to form a 30 nanometer structure. This is called 30 nm fiber of DNA. These 30 nm fibers make some loops like this. These loops are around 300 nanometer long. These are called 300 nm loops. After that, these loops can coil more to make 250 nanometer white fiber. Now I am going to the total picture. So this is our DNA. Each DNA strand is just 2 nanometer in width. In the DNA structure, we have found the nucleotide bases, and each and every turn we have 10 bases. That is the DNA. That DNA thread is attached to the nucleosome. Right now, the histone 1 protein is here. So this is nucleosome with the histone 1 protein, that is called the chromatosome. 2 chromatosomes are connected by linker DNA. Okay, this threaded structure is around like 11 nanometer width. Now all these threaded structures are coiled, super coiled here, making a 30 nanometer width of fibers. Okay, these 30 nanometer nanometer fiber, they are creating more coiling here, just look like structure. This is a 300 nanometer structure. These 300 nanometer structures, they are making more coils here, more supercoiling here. And that is creating that 200 nanometer white fibers. This is 200 nanometer white fibers. And this one, the width of this one is around 700 nanometer. And all this coiled into these 1400 nanometer width of chromosomes. This is the actual supercoiling in the chromosome. Highly supercoiled structures. So from here, there are a couple of things. Which one is very important? What is chromatosome? What is nucleosome? And this is a very common question. Tell us the nucleosome model of the chromosome. And this one was developed, that is first proposed by Sanger and Nicholson. Sanger and Nicholson's nucleosome model. Okay. Some chromosomes, they have different kind of patterns. You know, this is called polytene chromosome. And polytene chromosome is very, very important in case of studying chromosomes because it is very wide in structure and it has different kind of bending patterns here. This polytene chromosome is present in salivary gland of Drosophila. What is happening here? They can create by repetition and the replication of DNA, but after the replication, the cells didn't divide. So the replicated DNA is there inside the chromosomes already. And as a result, they are creating a very bigger structure like this kind of things. And in this type of chromosome, there are a lot of bands there, some area which are relaxed and creating some kind of bubble-like structure. Those are called chromosome puffs. Okay, this is a general structure of polytene chromosome, and it can happen. So there's some kind of changes can happen in the DNA structure. Another is that as the genes are there, and so they are coiled in such a way that is not permanent all the times. It can change with different environments. This one is one example of DNase 1 activity. When you will find the thing ending with "ase", that means an enzyme. Okay. Then if it is nuclease means which can cut the nucleic acid. Okay. If it is RNA nuclease, that is thing. If it is DNA nuclease, that means it is cutting the DNA. So DNase means deoxyribonuclease 1. That is the enzyme which can cut the specific part of DNA. Not all the parts. They have some kind of specific sequences they can cut. But DNase 1 can cut almost very common sequences there. So that's why it is a common enzyme. If you can cut in a particular sequence only, then it is called the restriction enzyme. Okay. So DNase 1 can digest the DNA, but that can depend on this DNA structure. This is the chick embryo. In the chicken DNA, it has found that generally in embryonic stage, they can produce the globin gene. When the globin gene is not produced, then this DNA segment is not sensitive to DNase 1. So DNase 1 cannot cut that DNA. But when the globin is produced at that time, in particularly 5 days of age of chicken embryo, this U gene production part that is becoming very, very sensitive to DNase 1. We don't know why. Okay. About 14 days of this chicken embryo, we have found that this alpha part and alpha B gene, alpha A gene, they are becoming very, very sensitive to DNase1, not the U gene. And in the brain cell, never it happens. The same DNA is there, but it never happens. There is no DNase1 sensitivity. It may be the production of globin gene can produce that one, but in case of brain cell, there is also the production of globin gene. We really don't know that why it is like that. But this is some kind of extra structure we can found in the DNA. Another is called epigenetic changes. This is the agouti locus in mice. This agouti locus determines the coat color in mice. If the parents have identical DNA sequences but different degrees of methylation, then they can create this kind of different coat colors. So methylation is not like that each and every DNA will get same kind of methylation. One DNA can get more, one DNA cannot get that much methylation because it is another extra addition of the methyl group into the cytosine. So if in the germ cell they got more kind of methylation or less kind of methylation, based on that one they can produce different kind of phenotypes. Now eukaryotic chromosome, there are two very important parts. One is called centromere and one is called telomere. In the centromere part, it is the constrictor region. Presence of kinetochore is there, only the attachment site of kinetochore protein, and this kinetochore is responsible for attachment of spindle fibers. Generally, in the centromeric location, there is no such active gene. It is highly heterochromatinized, and only function is the attaching of spindle fiber during cell division. And each and every chromosome, there is only one centromeric region. If it is, there is no such non-disjunction or other things. Telomere. Who is having the telomere project? You have the telomere. Okay. So this is one of the function of telomere. Telomere is the last part of the DNA. End of the linear DNA, that is called the telomere. Bacterial DNA is circular, so there is no telomere in the bacterial DNA. Bacterial DNA is like this. There is no telomere in the bacterial DNA. But eukaryotic DNA is linear in structure. This last part of the DNA is called telomere. Most important part is that in the telomeric sequence, there is one hang-up part, hanging part. Because during replication, this hanging part is not replicated. Always there is one hanging, because during this part, when we saw the replication, DNA replication requires one open OH group and the attachment of DNA polymerase into that group. In that part, they cannot be attached, DNA polymerase cannot be attached into that part because of highly repetitive sequences. They contain highly repetitive sequences, same sequence again and again, and it could be a 1 million times. Due to this problem, DNA polymerase cannot synthesize that part. And for you need to give this information. DNA polymerase cannot replicate that part. One enzyme is responsible for replicating the telomeric part, that is called tankyrase. Tankyrase and telomerase. These are two enzymes. Okay. In case of some eukaryotic cells and some mammalian cells, this open part, they can form this kind of loop. This is called the telomeric loop. Now the problem is that during each and every cell division, this part, this hanging part of the DNA is not replicated for each cell division. That means if it is not replicated, that part is off from the chromosome. It is being shredded off. So during our birth, what length of chromosome we had, right now we don't have that kind of length, because each and every cell division is getting shortened. And this shortening of chromosome results the chromosome open to some other chemicals, some other damage, some other mutation. Not only that, this is creating a kind of cancer. So the aging and cancer, it is related to this shortening of telomeric bond. And due to highly repetitive sequences, normal replication process doesn't occur into this one. Okay. Telomeres are, these are the repetitive sequences. Suppose in tetrahymena, this is the repetitive sequences of telomere. This sequence is there for 1 million times. All the times you will find that one. Repeat, repeat, and repeat. Okay. So each and every kind of vertebrates and that is eukaryotic chromosome, which is linear, they can have this kind of telomeric sequences. That telomeric sequence differs species to species, and the replication numbers also differs. Repeat number also differs. Okay. In some case, in some cell, even it is cell to cell. In some cell, you can find suppose 1,000 repeats. In some cell, you can find 10,000 repeats, something like that. Now, this another important thing, the amount of DNA. So right now, think that you can taking out the DNA from your body, from your cell, and you can weigh it in a mass machine, okay, mass scan. So you can find that there is some amount of DNA. Suppose that much picogram or that much femtogram. Generally, DNA we can find at femtogram level. Okay. Sometimes in nanogram level, if there is million of cells like that. So each and every organism have a particular amount of DNA, and it differs from organism to organism. This is called C value. C value of DNA is called the particular amount of DNA in a particular organism. That is the C value. Now, we have found that in case of eukaryotes, some DNA contains, some of the cells contain more DNA, some of the species actually contains more DNA into their cell. But we don't know why. Fruit fly contains about 35 times more DNA than E. coli. Human cells contains more than 10 times the amount of DNA from Drosophila, 10 times more. Salamander contains 20 times more than human. So we don't know why. This question is called C-value paradox. This is called C-value paradox. Why these organisms, these eukaryotic organisms contains more DNA? We don't know. This is C-value paradox. It has not been solved yet. If you can solve it, that is fine. That is the open question. There are some kind of question in math also. You know, I'm sometimes studying those things. In Pythagorean theorem, a square plus b square is equal to c square. Okay? Find some positive integer, in which case it will be a cube plus b cube is equal to c cube. You can try it when you have time. Positive integer. So it should not be decimal, it should not be negative. In such case, you will find this thing. This is a 350 years old problem. That's not been solved yet. So like this C-value paradox is something like that. We don't know why this much amount of DNA is there. And this is the genome size of that. That is, salamander contains this kind of base pair of DNA. We don't know why. And another very interesting thing is that later we will find during the protein transcription, protein synthesis, that we have around 21,000 genes, though our gene is 350 times more long than E. coli DNA. E. coli have 71,000 genes, but we have only 21,000 genes. We are creating 150,000 proteins, but E. coli is creating just 100,000 from 71,000. That is another paradox, we don't know why. Okay, some kind of other things which is called a sequence variation. We'll talk about this one later in our biotechnology class, which is called denaturation of DNA and renaturation of DNA. Denaturation of DNA is called the melting. So if the DNA is heated up to like 90 degrees centigrade, then the DNA strands are being separated. Okay, this is called the melting of DNA. This is called DNA denaturation. If you cool down that DNA up to 70 degrees centigrade, 70 degrees Celsius, then these strands, the denatured strands, can rejoin again, can heal again. So this is called annealing or renaturation. Using this process, we can copy the DNA in test tubes. And this is called polymerase chain reaction or PCR using this one. So heat up the DNA up to 90 degree, then add some primer, start the synthesis of DNA when it is open, new strands are formed, cool it down to 72 degree, then it rejoins, double-stranded DNA is formed, cool it down to 60 degree, it can heal up tightly, again heat it up to 90 degree, open it up, do the replication and do the same thing. This is called polymerase chain reaction. So if we can cut up this kind of DNA from there, a small 100 pp long DNA or 10 pp long DNA, we can create million copies of that DNA in test tubes. And in eukaryote, there are a couple of sequences. One is the unique sequence DNA. Generally, unique sequence DNA means this is a kind of gene. Okay, each and every gene is different. They have unique sequences, and they are not identical copies. And is the repetitive DNA. This is the main problem in bioinformatics right now. The repetitive DNA sequences. There are a lot of repeats. We don't know why. And repeats. Now, just think about this thing that when we are getting out the DNA, we don't know in which chromosome this DNA is from. We are getting out the total amount of DNA. We are cutting with restriction fragments, and there are a lot of fragments. Then we have found that okay, this is the sequence. We can run into the gel and we can find okay, this is the ATCG sequence of this part of the DNA. Now after this part, when we know the sequences, we need to match up the sequences. Okay. If you have the repeat sequences there, how can you match up? Suppose this is the repeat, and this is 10 million copies. You can find the one in one stretch of DNA, you can find the 5 repeats of this one. Another stretch of DNA, you can find the 10 repeats of this one. How can you add this? That is the main part of bioinformatics. That you know this is this sequence, you need to add this sequence with this sequence. In such case, sometimes we are doing the hanging part, that this part is common with the other sequence, so we can match up this part. So this one is starting, this one is ending, we can match up. So this is for this. So we can have the whole DNA sequence there. But if there are repeats, how can we attach the repeats? We don't know how many repeats are there. And that is the main problem in bioinformatics. That's why all the algorithms and everything is working on. They are trying to find out this kind of how to assemble these repeat sequences into the DNA. There are a couple of types of repeat sequences. One is called a tandem repeat, if the repeats are there side by side. That is fine. Interspread repeat. So repeats are there in some part of the chromosome, same repeats are there in the other part of the chromosome. Okay, we don't know. That is called interspread repeat. Based on this interspread repeat, one is called short interspread elements or SINE, SINE. What is SINE? This is a LU element. It is 300 pp long and present more than 1 million times, almost 11% of the human genome. This is a short type of interspread repeat. Another is called line, long interspread repeats, and this is almost 17% of the human genome. A particular sequence, about 1,000 million copies are there and containing 17% of the human genome. We don't know why. And there are some kind of repetitive sequences which is called microsatellite or satellite DNA. What are those repeat sequences? These are very small, less than 10 BP long, but present in hundreds of thousands to millions copies. And when you take out all those DNAs, cut into restriction enzyme and centrifuge, they can form a different pattern in the centrifuge tube. That's why they are called the satellites. And these are, as they are highly repetitive sequences, generally transcription is not possible into that part. It is rarely transcribed, and mainly this is present in centromere and telomere regions. These are called the satellite DNA. This is all about, in a very less way, about nuclear. Now there are two more DNAs. One is called the mitochondrial DNA and one is called the chloroplast. Okay. I will not go too much details into these two, the mitochondrial DNA and chloroplast DNA. But I put all the slides here so you can just go through it. I'm just going a little bit about them. Both are containing DNA, the mitochondria and chloroplast. These DNA, they are mainly circular DNA, and they are in a matrix of mitochondria and chloroplast. Do you know the structure of mitochondria or chloroplast? This is the structure of mitochondria. This is the outer membrane, and this is the inner membrane. This inner membrane is called the cristae, and the space inside the mitochondria is called stroma. In this stroma, there are ribosomes and the circular DNA. In case of chloroplast, these are called the grana. Each and every grana is made up of disc-like structures that is called the thylakoid. And between each and every grana, there is a space there, highly containing a dense fluid that is called the stroma. And one DNA is there, circular DNA, and the ribosomes are there. Now, if DNA is there and ribosome is there, what does it mean? Suppose I have found that in one cell there is DNA and ribosome. What does it mean? What can I guess from that? Any idea? No? What is the function of ribosome? Ribosome is called the protein synthesis factory. Okay. And mitochondria is called the powerhouse of the cell. Okay. So if the ribosome is there, that means there is a chance of protein synthesis. If DNA is there, that means there is a chance of formation of mRNA, and from mRNA, ribosome can create proteins. In these two organelles, ribosomes are there, DNA is there, and they can synthesize proteins by themselves. Their own requirement, they don't need from the nuclear DNA. They can synthesize their own proteins into these cells, these organelles. Based on this fact, we thought that sometimes they were protozoans or they were prokaryotes. They were the prokaryotic cells. They were not the part of eukaryotic cells before. This concept is called endosymbiont theory. What is endosymbiont or endosymbiotic theory? That means that once the eukaryotic cells were something like that, they didn't have any kind of mitochondria or chloroplast. Okay. One kind of bacteria were there who used to create this kind of energies, that is the ATP synthesis, and they have their own DNA, they had their own ribosome and everything. One of these eukaryotic cell engulfed that protozoa. That protozoa became, prokaryotic cell became the part of the eukaryotic cell, and that became the mitochondria. Okay, because all eukaryotic cell have mitochondria. So they engulfed that prokaryote first, the mitochondria. One of these cell containing mitochondria, they engulfed another prokaryote which were capable to do photosynthesis. Okay, so chloroplast, that is, they contain chlorophyll, which were capable to do photosynthesis. One of that eukaryotic cell, they engulfed this protozoa, and it becomes the part of this eukaryotic cell, and that is the plant cell. Not all the cells. All the cells have mitochondria, and only plant cells have chloroplast. Okay. So this is a theory. We don't know, but we are assuming that as both mitochondria and chloroplast, they have their own DNA system, they have their own DNA, own gene, and they can synthesize their own proteins. That means maybe they were prokaryotes before, and they were the part of the eukaryotic cell in this way. This theory is called endosymbiotic theory or endosymbiont theory. Okay. And these, they have some kind of unique characters. Uniparental inheritance of organellely encoded traits. Animal mtDNA means mitochondrial DNA inherited almost exclusively from female parent. So if you study the mitochondrial DNA, you can find out the mother, your grandmother, and such on the maternal line of the evolution. And we traced that, this mitochondrial DNA of human. We traced that one, and it went up to one female, just one single female, who was the mother of all these 7 billion population. And she is named as mitochondrial Eve. And her origin was in East Africa. So all the human population came from Africa, East Africa, and this, she was the only person which we traced up to that is called the mitochondrial Eve, using this mitochondrial DNA and maternal line tracing. Okay. And these cells, particularly the mitochondria, they have their own properties, and when they segregate during cell division, they can segregate in different ways. They can segregate in random, like this. That some kind of cells, they have two distinct varieties of mitochondria here, but when they are replicating in different ways. Some contains only the red mitochondria, some contains red and blue, some contains only the blue. We don't know why. But this is a maternal kind of replication of mitochondria, and it could create different kind of varieties. Some traits with the mitochondria is the petit mutation in yeast, some neurospora mutations, and in human cases, these are the diseases associated with the mitochondrial DNA. And in case of plants, is cytoplasmic male sterility. Just keep this in mind. And mitochondrial genome is generally this kind of structure, circular structure. We already found out all the genes from there with the restriction enzymes, and we know what are the genes there and what are the proteins they can synthesize. Don't need this one. Damage to mitochondrial DNA is associated with aging. So in case of aging, there is another thing is there. This is the most important part. Many human genetic diseases associated with mtDNA in the middle age or later. Oxidative phosphorylation capacity declines with the age. Those with the mutations on mtDNA start life decrease oxidative phosphorylation capacity. Okay. But this mechanism is unknown, and oxidative phosphorylation and oxidative stress is another part of the science to find out the relationship with cancer. I worked with someone with oxidative stress, and stress means any kind of stress, which can damage to the cell and which can develop some kind of cancer. Chloroplast genome, many traits associated with chloroplast, and that is the cytoplasmic inheritance. It is more or less similar to mitochondrial DNA, but they have different kind of genes. This is from the rice plant. They are chloroplast DNA, and we have sequenced all the genes here. And some sequences normally found in mtDNA, and they are in the nuclear DNA too. Some kind of this is the thing is that later we will discuss about this one, which is called transposable element. What is transposable element is that a part of DNA segment which can move from one genome to another genome, which can move from one chromosome to another chromosome, which can move from nuclear DNA to mitochondrial DNA, mitochondrial DNA to chloroplast DNA, those transposable elements, if they can move by themselves, then we can find these sequences in the nuclear or mitochondrial DNA too. Okay, and we have that kind of similarities there. So they have some kind of connection with the transposable elements between these three genomes. Okay, this is all about the chromatin structure. Mainly, the important part is structure of nucleosome, structure of chromatosome, linker DNA, and the high coil structure of chromatin, centromere, telomere, repetitive sequences. Okay. We have just 10 minutes. I'll start a little bit about our next structure, though it is not in your canvas yet. It will take a little bit more time. That's why I want to start today. This is the DNA replication. Okay. So right now, actual molecular genetics start. Okay. And you need to know this thing very well, each and every step very carefully, because it is required in later cases a lot. Okay. So what is replication? Replication is the formation of another DNA from the mother or parental DNA. Formation of another DNA. That is called the DNA replication. Now, if I think about that one, replication has to be extremely accurate. Each and every DNA should be, the new DNA should be the identical copy of their parental DNA. It should be that much accurate. Okay. So this is one example that one human zygote contains 6.5 billion base pairs of DNA. If there is only one mistake per million base pair, in 1 million base pair there is one nucleotide difference, you could have 6,400 mistakes. One nucleotide difference can cause a mutation. Okay. Later we will talk about the single nucleotide polymorphism or SNPs. The single nucleotide polymorphism, one example is that we cannot breathe very well at the high altitude. Okay, this is our problem because we are habituated with the sea level or just above sea level, about 1,000 meter. Okay, the Tibetans, they are living around like 6,400 feet high altitude all the times. They don't have any kind of respiratory problems. So what is the difference between these two? Our generation, that is our DNA or their DNA. What is the problem? Is there any adaptation? Then we sequenced both the DNAs. We have found that only one nucleotide is different between. What is the problem? Is there any adaptation? Then we sequenced both the DNAs. We have found that only one nucleotide is different between genomes. It is in the heterochromatinized part, and only nucleotide difference that could create this kind of differences. If there are 6,400 mistakes there, can you imagine how could be the difference? Probably there is a new species, something like that. So it should be extremely accurate. And one example is that E. coli contains about 4.6 billion base pairs. And if that replication rate is 1,000 nucleotide per minute, 1,000 nucleotide per minute, then it would take 3 days to replicate. But E. coli replicates in 20 minutes. Each and every 20 minutes they can replicate. The speed is 1000 nucleotide per second. In 1 second, they can replicate 1000 nucleotides. This is high speed, high speed. In case of human, this is more. In human DNA, they can replicate in 24 hours. Total human cell cycle. Actual, the replication time is 4 to 6 hours. In that phase, they need to replicate 6.4 billion base pairs. Okay, so it should be more accurate and it should be in high speed. And we have all the mechanisms for that. Generally, in a nutshell, today I just want to give you the idea about that one. All this replication process is done by different enzymes. Okay, one enzyme is there who can read this DNA sequence and add the nucleotide for that. Suppose they read that this DNA polymerase 1, it is going to the DNA strand. It is reading, okay, A is here, so I need to add T here. C is here, I need to add G here. G is here, I need to add C here. Like this. Now, this function is done for 1000 nucleotide in 1 second. So there is a chance that enzyme can do some mistake. Another enzyme is there just behind it who can read the proofs, whether the attachment is correct or not. That is the DNA polymerase 1. That function was generally in eukaryotes, that is the DNA polymerase epsilon and DNA polymerase alpha. He is reading that one, okay, that person did the correct thing or not. Proofreading, that is called the DNA proofreading. They can read whether the sequences are correct or not. If the sequences are not correct, they can cut out that nucleotide and add another nucleotide, the perfect nucleotide, into that area. Okay, so this is the function they can do in our cells. That's why it is accurate and it is fast. Now the question is that this is DNA. So how can DNA be replicated? Based on this concept, there are 3 things here. So this is our parental DNA. Now the question is that if the new DNA is formed like this, then this is parental DNA, another DNA is formed. Or this DNA can be broken up into pieces and new DNA are formed here. Or there is a chance that these 2 strands are separating, the parental strands are separating, and new DNA is formed here. These are 3 concepts. In case of first concept, the parental DNA is here, another new DNA is formed just seeing this parental DNA, because we need the template DNA. Anyway, this DNA from which the new DNA is formed, that DNA or the parental DNA is called the template DNA. From this template DNA and new DNA should be formed. So this is one pattern. This is called conservative mode of replication. These are all hypothetical. Okay. Next, the question is that the parental DNA strand or temporal DNA strand is broken into pieces and the new DNA is formed there. This pattern is called dispersive mode of replication. Dispersive mode of replication. Third pattern is this, that there are 2 strands of DNA, they are getting separated and the new DNA is formed along these strands. This is called semi-conservative mode of replication. Clear? So there are 3 hypothetical patterns because we didn't know that what is the pattern of DNA replication. Either it is conservative, or it is dispersive, or it is semi-conservative mode of replication. So we need to prove that one. To prove that one, this experiment is very, very important. Messenson and Stahl, they did this experiment. What is the thing that DNA contains nitrogen? Because all the bases are nitrogenous bases, right? So nitrogenous bases means they have nitrogen there. So they added 2 types of nitrogens. One is the 14N, that is the common form of nitrogen, another is 15N, that is the radioactive isotope of nitrogen. They prepared culture medium with this 14N nitrogen. So at first, and another culture medium is the 15N nitrogen. Okay. They cultured the E. coli in the 15N media first. That means all the nitrogenous bases into that E. coli contains that 15N nitrogen. Clear? That means if you found this E. coli, you will find all the 15N nitrogen there. Then he cultured that one in 14N media. What does it mean? If the new DNA, when the new DNA is formed, all the new DNA would contain that 14N nitrogen. Okay. So the DNA one would be, if it is conservative, then this DNA is 15N, the new DNA is 14N, if it is conservative. If it is dispersive, then one part is 14N, other part is 15N, another 14, another 15, like this. Okay. If it is semi-conservative, then one DNA is 15N, the parental DNA, because it was cultured first in 15N media. One DNA should contain all the 15N part. Another DNA, the new DNA, would contain the 14N. Right? So this is our initial stage of experiment. Now, how can I know that it contains that much amount of 14N DNA, that much amount of 15N DNA? The thing is that you need to take out the E. coli from this culture media, then centrifuge it. When you centrifuge, you will find the density gradients. Based on the density gradients, all the DNA, they will create these two types of bands. 15N is heavy, it will go to the down. 14N is little bit less heavy, it will go to the top. So you can find these two types of bands. One is 14N band and one is 15N band. Okay. I think I will go. Now, if it is the conservative mode of replication, after 2 cycles, you will find this is our parental DNA, that is a 15N media, 15N DNA. The new one is 14N. So these two DNA will form the new DNAs. So we will find 3 14N DNA and 1 15N DNA, right? After 2 cycles. In such case, if I centrifuge that one, I'll find this kind of binding patterns. Okay. So 1 15N, because 15N is very less here, and the 14N, that is little bit thicker because 3 14N DNAs are there. If it is conservative. If it is dispersive, all the times they are mixed. 14N and 15N are mixed here. Okay. If I centrifuge all those things, we'll find only one band. That is a mixture of 14N and 15N, and that should be between these two. Between these two areas, because that is a mixture of 14N and 15N. That DNA. If it is semi-conservative, in such case, what will happen? The first one is 15N, parental DNA. The new DNA is formed around the parental DNA, they are separated. So that is a mixture of in the first generation. In second generation, they will separate. So there will be one 15N, one 14N. In 14N, they will create another 14N structure. In 15N, they will have the mixture structure. Okay. In such case, we will have a mixture band at the middle and one 14N band on the top. Try to understand this one. Clear? 3 different bending patterns can tell us what kind of mode of replication is in the DNA. This is hypothetical. Did the experiment, they have found the first one. Okay, that is this one. This kind of bending pattern, which tells us that DNA replication is in the semi-conservative. So there should be one parental DNA. Around the parental DNA, the new DNA is formed. Okay, this is the Nathan-Stahl experiment. I didn't put this slide yet. I tried to finish only this part up to today because next day we'll go for the whole molecular level. This DNA replication, I went through your book. It is a little bit different in your book. I will add a couple of more pictures there, and my slides will be enough for that one. And other than that, you can read your book. Okay. Real molecular biology.